Thank you. This is a non-commercial talk. I'm from Cybergenetics, but I won't be talking about TrueAllele, which is the system that implements much of what you're seeing here. Instead, I'll be focusing on the science and the background, how it's used, cases, and validation studies. And the focus today is taming uncertainty in forensic DNA evidence. So first, what is uncertainty? Well, people really crave certainty as something they can have, but inherently, scientific data are uncertain. If you want certainty, you have to go to religion or some other field. Probability, though, has been a standard way for over 100 years for scientists to describe uncertainty. And sort of the converse of uncertainty is information, and as we'll see, and as you already know, information can be described as a change in probability, which is a very powerful way of looking at it, particularly if you have different probabilities of uncertain events and you want to quantify how much information was gained. The approach that I'm taking is that DNA identification is an information science, and that the goal of this information science is to obtain the maximal objective information possible from the evidence that we're provided. So I'm going to describe an approach to how to look at forensic DNA in which there are only two concepts. One is the genotype, and I'll describe it in a way that some of you who are more statistically minded might be familiar with, but you've seen, as you'll see in the example. So if we have a genotype of an unknown person, there's a question mark in the box on the left, and we ask, what is that person's genotype? We've obtained no data yet except maybe the fact that they're human. And so all we know is our prior belief, the prior probability, that they belong to some human population. So all that we can say is the genotype is some particular set of allele pairs up to probability, as determined by the population. And I've represented in the middle different bars representing degrees of probability for different allele pairs. So that's what we know as our prior probability before we see data. Now we see data. And for the moment, we'll take a look at something you're very familiar with, which is paternity. And this is kinship data. So in blue, we now have information with arrows coming out of the unknown individual, the true biological father. And that arrow is representing a likelihood function. What a likelihood function does is it says in order to explain the data, in this case the child, which has been genetically inherited, one can try out all possible values of allele pairs that might explain that data. So in a non-mathematical sense, the best way of thinking of a likelihood function is how it explains data. And in order to explain the data, you would see in the middle a likelihood function that would have the usual 0, 1, 2 that you see in genetics, in kinship, that's trying to say that some allele pairs are more likely than others as a way of explaining the data, which will be shown in blue throughout the talk. And as you're all familiar with, if we have a prior probability, the population flow in, shown in brown, and we have a likelihood function, which is the flow into the data coming out of the questioned individual, out of the biological father, we can form a posterior probability. And that's done objectively with Bayesian inference. And it's done by saying that the posterior probability, which is this renormalized product in the center, shown as a probability distribution of bars of different probability coming out of the allele pairs, is proportional to the prior probability times the likelihood. That is what you believed before times the impact of the data. That's what a likelihood function does. The reasons that it's objective is if you look at the picture at this point, we're not comparing it against anyone. There's no suspect. There's nothing. It's been inferred completely from data. And the reason it's Bayesian is that Bayesian inference is the only valid probability method I know of where you can combine the belief before and the data that you see in order to obtain a valid probability distribution. In this case, our belief before had nothing to do with who anyone was. We don't know that. It had to do with the human population, which we can scientifically measure. Now we enter forensics with a second concept. So the first concept is genotype, and throughout this talk, or perhaps the rest of the century, genotype will mean a probability distribution whenever there's an uncertainty in an allele pair. But for forensics, we now need to compare genotypes, and so the second concept is match. Once you identify a particular individual, or a database of individuals, you now can compare the allele pair, the example here is 1011, with something that might have appeared with non-zero probability in the probability distribution. Suppose that 1011 allele pair had probability 50%. Then you can see that the probability that the genotypes match, if it's 50% from the evidence and it's certain from the putative father, would be 50%. That's the strength of match. But that's not forensic science yet, because we need to know how rare that match is. What's the extent of coincidence? And so to do that, we bring the population back in, and we now compare on the bottom that candidate putative father with his 1011 allele pair back against the population. Suppose that the population coincidental probability or prior probability is about 5%, shown by the thickened brown bar here in the center on the bottom. Now, the probability of a coincidental genotype, say in this example, is 5%. And so now we have a balance of two probabilities. We have the matching genotype probability in the numerator with the evidence, and the coincidental genotype in the denominator. And when you have that balance of probabilities, and you take the ratio, one of the many forms of writing down the likelihood ratio, this paper describes about 4 different forms. This one is particularly intuitive to forensic scientists, the notion of a matching genotype and the probability of that event, divided by a coincidental genotype, the probability of that event. We have a likelihood ratio. There are many formulas for likelihood ratios. They all give the same answer, but some can be explained more easily than others in different contexts. And so if we take that 50% probability of a matching genotype and divide at this locus the 5% probability of a coincidence, we obtain the ratio 50 over 5, or 10, and that is the paternity, the match statistic, the likelihood ratio at this locus. Now, fortunately for forensic science and the log of the likelihood ratio, the orders of magnitude or powers of 10, is a standard measure of information in most fields that measure information, having nothing to do with forensic science. And so in this example, if there was an information gain of 10, the log base 10 of 10 is 1, 10 to the 1 is 10, and that's one information unit. And that's the unit that people who develop methods like Peter Gill, John Buckleton, Bruce Weir use in papers when they compare efficacy of methods, and that's the unit of information, the log of the likelihood ratio, we'll use throughout this talk. So if you take the same approach, but we try to tame uncertainty the way scientists do by looking at more data, you see that the kinship problem is no different. We still have an unknown individual, in this case a missing person, it's no longer paternity, with a question mark indicating we don't know the genotype of that individual. And suppose in addition we add another offspring, maybe a daughter, there are other relatives, maybe a father. What you tend to see is a sharpening of the probability distribution to whatever the data is trying to indicate. And so here I've suggested on the top that the probability maybe went from 50% to 90% as more information. Again, in blue, that's a joint likelihood function that involves 2 children as opposed to just 1. And therefore, the probability of the matching genotype in the numerator almost doubles. The coincidence probability in the denominator stays the same, so the likelihood ratio would double, since the likelihood ratio is the ratio of those 2 probabilities. So that's the general approach that we'll be taking to DNA forensic science. There are 2 things that we care about: genotypes, known up to probability for uncertainty, and matching in order to make a comparison and form likelihood ratios. So now we'll bring in mixtures and talk about phenotypic data instead of purely genetic data. The concepts are identical. The only shift is instead of using genetic offspring for likelihood functions, we'll use observed data for our likelihood functions. The math is the same. It's just a matter of how you make use of the data. So suppose we have a mixture, and in reality there are two people there. The happy face maybe is the assailant, and the unhappy face is the deceased victim, something like that. And that corresponds to reality. There really is some state of the world where there are these two genotypes that are residing in the evidence sample. Well, we can't tell by looking at it. At this point, all we could talk about would be the population or the prior probability. So we go to a laboratory, and I'm going to sum up everything that you do in the words LAB, lab, and the lab produces STR data. Okay, and that's the starting point for any interpretation, whether it's by computer or by eye, is the electronic STR data, printouts thereof. And what we now have in that data is an observation. Of course, the observation of data is not the same thing as reality. I could go into Plato's cave, or seeing shadows, all that sort of stuff. But I told Ingo I wasn't going to do all that, so I won't. So we have observations which are philosophically different. But from that data, we can make inferences. And all methods of mixture interpretation, whether the practitioner actively writes down the allele pair probabilities or not, produce probabilities of allele pairs. Every list of allele pairs, incidentally, it corresponds to a likelihood function. You combine it with the prior of a population, and out comes your match statistics for LRs or RMNE or whatever. So this is quite general in that whatever you do with mixtures, you're going to infer something that's equivalent to a genotype with probability according to some model. And philosophically, that model is trying to understand reality, but it's not the data. It's not the observation. The genotype is not the peaks. It's not the alleles. It's a statistical inference that we make from the data. These are three different levels of reality. So the model is exactly the same. With phenotypic data, we still have on the top left evidence with a prior coming in with a brown arrow. We want to know a genotype. So in blue, we're showing there's data, in this case phenotypic data, as opposed to genetic offspring data. And a probability distribution is produced. In this case, I'm indicating in the probability distribution on the top in blue with a thick bar at 10, 11, maybe the genotype allele pair of that second contributor is about 50%, just as a running example. And when you compare that with a known person, remember before we did that comparison, it was just a probability distribution. It's completely objective. We had no idea who we would be comparing it against or what million people we'd be comparing it against. The instant we decide to make a comparison, that allele pair selects out one of the allele pairs with some probability. And that gives us the numerator, the probability of the matching genotype. And we have the same coincidental genotype in the denominator. I'm not going to go into population substructure, theta corrections, and so on, but you can. It's more detail. And again, you obtain a likelihood ratio, which is a ratio of the 50 over 5, which would be about 10. So one log unit. So it makes no difference where the data come from, kinship, inheritance, mixtures. The concept that we have likelihood functions and the data is taming our uncertainty is statistically and mathematically is the same. So to give you some sense of relief, we're not expecting that there'll be training courses where you learn to do all this by hand, which you may have been concerned about. Instead, we're really talking about computer interpretation, where you set up a problem, you give it the data, whether it's kinship data or mixture data, low template DNA, whatever, and then you let a computer run quantitative computer interpretation on it in order to infer genotypes. These sorts of computer interpretation systems involve a statistical search of a probability model. We'll talk not too much about any of the math of the models, but you have to do a very complete, thorough search to use Bayes' theorem. You have to explore all possibilities. The goal is to preserve all the identification information present in the evidence. And if you infer the genotype objectively without ever seeing a suspect, then it's objective. And then the computer is required to record in a database or in a program what it found, and only afterwards is any match made. So it can't have any examination bias. The systems that we use can use any number of mixture contributors. I don't think we go much beyond 5 in routine work. They model most everything in the STR experiments: stutter, peak imbalance, degraded DNA, mixture weight, so on. And most importantly, in order to not use thresholds, they have to calculate the uncertainty of every peak. If they don't do that, then you don't know to what extent you can rely on the answer. But since a classic paper by Gelfand and Smith 20 years ago in the statistics literature, I'd say for the last 10 years, most any serious or publishable statistical paper that does this type of reasoning spends a lot of time working out the uncertainty of the data. So this is what quantitative data look like, which you're familiar with. This is a two-person mixture. There are some higher peaks, some lower peaks. And the task is to explain the data that are in front of us. The starting point, if you see instead of the green curve, you're now looking at these gray triangles. That's one explanation. You assume, say the taller peaks are one allele pair, the shorter peaks another allele pair. On the bottom we see some stutter modeling. There's some relative amplification. There's a mixture weight underlying all the loci corresponding to the template. That would be one way of saying with these particular genotype values and other experimental parameters, this is one explanation of the data. This is an explanation that might fit the data pretty well and have a higher likelihood than one where the explanation of where the alleles were didn't correspond to where the peaks were. Something else that's important is calculating the peak uncertainty. Peaks are not just peaks. From a Bayesian probability perspective, your data are random variables as well. And so every peak has a mean and a variance. And so I've represented the center of that peak is where it's most dark, but that gray is trying to show a probability distribution of where the data might be. This is equivalent to if you took some template DNA and you amplified it 30 times and you looked at what the peak variation would be. Computers can do that from data as well. You don't have to do that in the laboratory. And something you'll also notice is that the uncertainty scales with the peak height. The spread increases linearly with the variance, and the relative certainty decreases. But once you know where the data are, and the computer knows where the data are and what the uncertainty is, then it can begin to try out every possible explanation, including the genotype. So what I've shown here is maybe one individual at this locus, indicated by the two short blue alleles, and another individual might be homozygous at this locus in an orange allele, so this two-person mixture. I'm not showing all the other variables like stutter and so on. And what a computer must do in order to give an accurate answer is to try out every possibility of how much DNA there is, different alleles, and so on. If your alleles are located nowhere near the data, if they're not falling into these probability regions of gray, of the probability distributions, but they're at the wrong location, or they're too high or too low, then the probabilities of those patterns, as determined by the likelihood function, will be 0 or negligible. As you get into the region of where the data are, you get more and more probability, the likelihood gets higher. And then when you're in the region, as the computer searches, of explaining the data, could be many different sorts of allele pairs that could do it, but it looks like you're now starting to account for the data. That's where you get higher likelihoods and more support for the hypothesis that this is one of the proper allele pairs to be considered with some probability for a contributor at a locus. There are other approaches that you may be familiar with that involve thresholds. Show of hands. You all use thresholds, that's fine. The qualitative thresholds have a different approach. The idea is that peaks that are over threshold are treated as allele events. Peaks that are under threshold essentially maintain that the alleles don't exist. And then the data are reduced to all or none events. Either it's there or it's not there. And you end up with a list of the included alleles with the quantitative information stripped away. This is in many interpretation methods. There are some human methods that do use peak height data. But the concept is either the alleles are there or not. If you've made a good guess as to that those are really the alleles that are there, even though in Bayesian and modern statistical reasoning you don't ever change the data, this might have scope. Of course, if there's an error rate associated with thresholding peaks to find alleles, that could be problematic. And certainly, as John Butler showed in a famous or infamous study 5 years ago, the MIX-05 study, when I think it was over 50 laboratories were sent the same two-person mixture with a known victim from NIST, the variation, the consistency between the laboratories ranged from 4 log units, 10 to the 4th, to 14 log units, about 100 trillion, and it was all over the place depending on the particular methods that were used with thresholds. So some methods have more efficacy than others, up at 100 trillion, others down at 10,000. And certainly there is a range of variation when it comes to the reproducibility of human interpretation of threshold data. I'd like to now go into some examples with that background. So this is a case, as far as I know, it's the first case in which a rigorous Bayesian interpretation with a thorough search, the computer spending hours trolling over the data, trying out every possibility of allele pairs, stutter, and so on, was done. And this was in Pennsylvania. It was done 2 years ago. And what was striking in the courtroom in the U.S. is that really nobody had ever seen the notion of more than one match statistic before. This is totally alien. If you get a statistic, if the crime lab comes back, I'm going to use the word inclusion to mean RMNE, CPI, and CPE, but I'll just use the word inclusion because it means all those things. They're just used to the lab coming back with a statistic, typically inclusion in the U.S., and that's the match statistic. The fact that an outside expert used the victim, that was 93% from this fingernail evidence, and obtained a match score that was 3 log units of likelihood ratio greater was a bit surprising. And the fact that a computer used the quantitative data as well and obtained something that was 7 orders of magnitude larger was very unsettling, and basically all the defense could talk about is that DNA interpretation must be highly unreliable with mixtures if you get different answers with different results. So the prosecutor did a good job, the voir dire went well, and he was convicted. But the concept is the probability modeling preserves the information that the peak thresholds discard. Last week, the appeal came through the courts in southwestern Pennsylvania, and I had the pleasure of observing the prosecutor and the defense. The defense went on again for the appellant, saying, well, there are 3 different statistics. Mixture interpretation and DNA must be unreliable. The prosecutor had done an excellent job of reading the literature. He quoted from the ISFG study by Peter Gill et al. From 2006. And his closing line at the trial was that the inclusion method is not the preferred method of mixture interpretation, which is probably the first time in a U.S. courtroom a prosecutor said that. It was last week. But you all know that because it's been out there for a while. So I want to move to another case and start illustrating how in science we tame uncertainty by using more data. This is a case where there was low template, very low template DNA mixture. There were believed to be 3 DNA contributors. The amplifications were done in triplicate, and then a post-PCR enhancement was done. So those peaks that you're seeing with peak heights of 100 or 200 are really 10 times higher than what you might have seen on the original electropherograms. Human review found no match statistic, which is why computer interpretation was requested of the evidence. And instead of using a likelihood function, like when you have only one piece of data, like one offspring, as in Kinship, when you use 2 or 3 or 4 children, you can use multiple data traces. In this case, there were 3. This is not at all the consensus method where analysis is done and then things are put together afterwards. Instead, the inferences are made as the hypotheses of the different genotypes, stutter, mixture weights, and so on are trying to explain independently all 3 items of evidence simultaneously, which is the statistically valid way of doing it if you do statistics. There's a product of likelihoods, so these 3 times the prior, that's what gives you the posterior distribution. What we found, let me give an example at VWA. So in blue, you see the genotype probabilities. I mean, if you look at the original data, each of these patterns looks different. Different amplifications, very low-level DNA, a lot of variability. And there's a probability distribution that's produced where if you looked at it, you'd say, well, it's clearly not indicating any one particular allele pair at that locus for that unknown contributor. Shown in brown is the population distribution of the prior, what was believed before. But it doesn't make any difference because the way you contain uncertainty by using more data, but then once you have your probability distributions, before in brown and after in blue, you go and you ask, who am I comparing this to? Right? The computer has no idea what your question is for the match comparison yet. It's incapable of knowing that. So you identify that you're interested in 1418. And you say, well, suppose that's the suspect here. Well, then nothing else is relevant by Bayes' theorem of the likelihood ratio. The likelihood ratio is a two-state variation of Bayes' theorem. All that's relevant is the posterior probability divided in blue divided by the prior probability at the suspect's allele pair, 1418. We see a 6-fold increase in probability. That's all that's relevant. And when you multiply these small probabilities across all the loci, accounting for population substructure and the number of contributors, the match statistic is over 3.5 million. And that's what was described in this case. We're going to give one last example from a case, again showing that more data can give more information to tame uncertainty by using joint likelihood functions and combining data in a rigorous way. Here's a handgun where there were 4 swabbings made and 2 amplifications indicated by the little stars from each area that was swabbed. This is what the data looks like at locus D18. For each swabbing area, we show the 2 amplifications in blue and in green. And if you look at each amplification, if you can see the data, the amplifications look fairly similar from each swab, but they look a little dissimilar across the 4 swabs. They have different patterns, they're different swabs. In this case, what we were interested in finding out was how much information could we get about the minor contributor that ranged from 20 to 40%, depending on which swabbing it was. And these are the results that we presented at the Sydney conference last year. If you look in light blue, next to the number 1, that's looking at each item of evidence independently in isolation, just one at a time. You get a range of different log likelihood ratios, which is our measure throughout this talk. And you're reaching the point where one of those gave reasonable information, maybe around 10 to the 8th, which would have been enough for government work, but this was more of a research question. The original lab made no statement at all about the minor contributor because with the guidelines they had, there was nothing that they could say. In fact, we were actually asked to look at the major contributor, but the computer found that uniquely for all 8, so we didn't pursue that as a research question. Now if we combine the 2 amplifications, if you see in green 2, we combine them in the sense of a joint likelihood function. The computer must consider both EPGs of data simultaneously, and we see that for some of those items, we get a gain of information of about 1 log unit. Now, we tried out all 6 ways of combining 2 items, or 2 swabbings, together with their 2 amplifications, that's shown in orange. And on the bottom row, you see we pick up yet 1 more log unit out to about 10 to the 12th. And then using all 8 amplifications together in a joint likelihood function with all 8 items of data, we obtain essentially the full random match probability and an essentially unique minor contributor genotype. So I'm going to move on now to validation studies. This was a study that was reported 2 years ago. It was started as a National Institute of Justice grant 10 years ago. The data were generated by Margaret Klein at NIST and John Butler's lab. And let me first tell you what the blue dots are. The experimental design was to take 5 different mixing proportions of two people in 90%, 70%, 50, 30, 10%, so different mixing proportions. 2 different pairs of individuals were used. And then for each pair of individuals and each mix of the 5 mixture proportions, 4 different quantities of DNA were examined: a nanogram, half a nanogram, quarter of a nanogram, and an eighth of a nanogram. And so each of those blue dots corresponds to one of those 40 experiments as they've been interpreted by computer. In the paper, we did it without assuming the victim was known, with assuming the victim was known, under different methods. In this particular case, for comparison, this was done assuming the victim was known. And then for each of those points on the scatter plot, on a log-log plot, we look at 2 variables. One is the quantity of DNA from the unknown culprit, and that was easy to figure out because you multiply the known mixture weight from the experimental design times the quantity of DNA. That's the amount of DNA in the unknown fraction. And then the y-axis is showing information, the log of the likelihood ratio. And what we see on this plot, proceeding from the right, so it's on a logarithmic scale, at the end we have 1 nanogram. Here's 100 picograms. On the left is 10 picograms. Over the 1,000 down to 100 picogram range, the computer is recovering, using the victim on a 2-person mixture, essentially all the match information that was present. And then, as it decreases from 100 nanograms down to 10 nanograms of the unknown fraction, we have a linear regression line that accurately models the loss of information. And if we consider 1,000,000 to 1 to be a convincing threshold, it crosses at about 15 picograms. In red, we're looking at the computer's running of an inclusion method. And as you would expect, there's a shift to the right of 1 order of magnitude, where RMNE, or inclusion, has a sensitivity to produce 1,000,000 to 1 match score of about 150 picograms. So there's a loss of information. It has its own regression line, and most of the dots, all that were appeared in blue are not appearing in red as more and more information is being lost. Okay. This is another study that's coming out in Journal of Forensic Sciences in November, done in collaboration with Barry Duesman in the New York State Albany Laboratory. We looked at 60, there are many parts to the paper. It's a long paper. People who've read it here have told me it's taken them 3 days. But there's a lot in it. It has the whole theory. It has a very detailed example on 1 item showing at 10 loci, 10 different reasons why the match information dropped from 1,000,000,000 to 1 down to 1,000 to 1 with RMNE. And then it has the larger results on more cases. 8 cases were reported out as 2 unknown mixtures, and another 8 were reported out with the victim known. In this case, since I'm comparing with inclusion, this is 2 two unknowns, and we don't know the victim. For each item is shown on the x-axis, and the y-axis is showing the log of the likelihood ratio as an information measure. What you see in the blue bars across these 8 cases is quantitative interpretation produced an average match score of about 10 to the 13th, about 10 trillion. The result that was reported using CPI, the inclusion method, in the case folder was fairly typical. It's usually in the US with 13 CODIS markers, about 1 million to 10 million. Here was about 10 million across the cases. And if you look at the gain in information, it's about 6 log units, about 1 million to 1. And that's what we've seen in hundreds of mixtures like this. It's what we'd expect. Sometimes if it's a 50-50, the gain is only 2 or 3 orders of magnitude. If it's a more informative 70-30, you might get 10 orders of magnitude gain. But on average, it's 6 orders of magnitude. We also looked at the reproducibility, since in addition to knowing how efficacious, how well a method preserves information, it's also important to know how consistent it is. These are statistical programs, so they're not expected to sample in the same way twice. But the variation that was measured within group, if you see in the blue and the green bar, those are 2 replications by the computer on the same data. The numbers are reasonably close. The within group standard deviation is measured in tenths of a log likelihood ratio information unit, which I would say compares favorably with 100 times greater variation in the US community with thresholds that John Butler had found 5 years before, over 10 orders of magnitude. This is another study we're doing on the same data. We're writing up now with the New York lab. Here what we looked at were not just the mixtures that were selected for having a human review score. We looked at all the mixtures. And here are all 86 items across, shown on the x-axis. The blue is showing in descending order the information, the log likelihood ratio of how much information was present in each of those cases. The median value is 10 to the 15th, about 1 quadrillion to 1. And the area represents the full extent of information that was found over the study. The bars superimposed is the human review. There were 4 in gray, assuming it wasn't a mixture using RMP. There are a few CLRs shown in green where the victim is used that had roughly twice the information as the lower level bars shown in orange, which was inclusion, which tended, as expected, to lose most of the information. The most telling statistic here is of the 86 items, fewer than 30% were given any match score at all by human review. So 70% of the evidence was not quantified and wouldn't be usable in court in many jurisdictions. And then if you look at the total amount of information gained, the ratio of the areas, the human review with thresholds produced less than 20% of the total information that was present. We've done more refined studies to identify how many cases received no match score at all. It wasn't that they found a result and then they stopped trying. They tried on everything, and many cases have no result. Other cases have multiple results. And so again, the quantitative interpretation using all the data, using probabilistic genotypes, considering all the possibility, tries to preserve all the information, whereas using peak thresholds tends to discard it. So you may be asking, why is it that thresholds don't perform as well? Is that a question you have? Okay, so we'll try to answer it in the next few slides. And there's more in that JFS paper. So the peak height of the data in EPGs is a random variable that has a mean and a variance. Let's take each separately. Because it's a mean, it suggests that there's a probability distribution of what the underlying quantity of DNA actually is, as reflected by the peak that you happen to observe at the center of that probability distribution. So if you set a threshold that goes in between or on one side or the other, you're taking a perfectly happy probability distribution that is only telling you likelihood and probability and has no concept of error, and you're artificially introducing error. Either you made the right decision, but that's where the mass was, or you made the wrong decision. There's no decision to make when you use probability distributions. It's just there. The second component is subtler and has to do with the variance. Many papers over the last 20 years have shown that with STR data, following the mass laws of chemistry, the variance is proportional to the peak height. That is, as you increase how much DNA there is, if you double the DNA, you double the variance. If you quadruple the DNA of the peak height, you quadruple the variance and double the standard deviation. Flipping the ratios, the relative uncertainty gets better as the amounts go up. However, thresholds implicitly, when you set a stochastic threshold in your lab, you're choosing or identifying a number like 170 or 200, 250 RFU, and that number is built on a model that assumes a constant variance. It's just the wrong model. That isn't how data behave. They don't have a constant variance. Most of the variation that you see in your data doesn't come from the detection instrument. It comes from the amplification in the PCR. And PCR follows a mass action law. And so the constant variance assumption of thresholds and stochastic thresholds is just wrong. There's no basis for it. And if you put it into a computer and you model it both ways, you start getting very bad answers by assuming the constant variance that no one has ever observed when they've done repeated STR experiments or modeled what PCR does. So at some level it can't work because the underlying model's wrong. What happens in practice, if you look in dark blue on the left, there's a probability distribution, and you see that the evidence is pointing toward one particular allele pair out of the others. The methods with thresholds start allowing many methods the data doesn't even permit, particularly RMNE. When you look at it, you sort of realize that with the peak heights, those aren't even feasible answers. And the result is you diffuse your probability, disperse it over many events that are either unlikely or not possible at all. As you diffuse away from where the probability is, you're lowering the probability of where it is at the true allele pair, with the result that since your match statistic, the likelihood ratio, has as its numerator the probability at the matching allele pair, you just keep reducing it by diffusing it into impossible events. So that's the harm that it does by considering things that really are impossible and giving them more probability. This is a study that we're writing up now where we took the same NIJ data provided by NIST on the 40 mixtures, and we had people in triplicate, just to detect any errors, apply 3 different thresholds to the data at 50 RFU, 100 RFU, and 200 RFU. And we looked for an error rate. And the error rate we're looking for is a false negative rate. What are we missing? And that's measured in the number of alleles per locus. You see that if it's a 90% mixture, over here on the left, then with the bars in order are going 1 nanogram, half, quarter, and eighth of a nanogram. But you only have a 10% error or so, a false negative, if it's a 90% mixture, all the way on the left, with 1 nanogram. But as you go to half a nanogram, those heights on the left cluster start going up, and they soon exceed 100% error rate. When you get to a 30% minor contributor, which is quite common in practice, the error rates start going up. You're up at 40% missed alleles per locus, even with 1 nanogram, and it rapidly goes up to over 100% error. And when you have a 10% mixture, which is the sort of cases we get sent to look at, but even at a nanogram, you're already over at 100% error of the missed alleles. I mean, if you think about it, if you draw a high threshold and there's low-level alleles in a 10 or 30% mixture, you're not going to detect them. And that gives you a high false negative error rate. And I don't know of any other field of science where we routinely see false negative error rates exceeding 100%. So preserving information is quite important, and the whole approach that I described, that forensic DNA, whether it's kinship or STRs or low template DNA, it's all the same approach. We infer genotypes up to probability, and we match them and produce information statistics, likelihood ratios. So now imagine instead of having one item of evidence and one suspect, we have a set of evidence genotypes, a forensic database, hundreds of thousands of genotypes. And on the other side, we have a million suspect genotypes, convicted offenders, arrestees, whatever your statutes are. In the same way that one can preserve these genotypes when analyzing a case and then form a likelihood ratio by plugging them into a formula, an investigative DNA database can do exactly the same thing. Instead of looking at allele sharing or heuristic methods to see what might be there, high stringency, low stringency, whatever, it just produces a likelihood ratio. And to do that, you need a genotype probability representation. Alleles won't do it. The alleles are basically RMNE. Any representation in a database that's giving alleles is doing exactly what inclusion does and throwing out information to the same extent. But genotype probabilities can fully preserve the DNA identification information and enables a true likelihood ratio calculation with every match that comes through, and lets you do all the things you expect to see with DNA databases, connecting crimes to criminals, disaster victim identification. We use the system to reanalyze all the World Trade Center data, for example, including kinship, victim remains, and personal effects, to find missing people, conduct familial search, or other sorts of homeland security activities with DNA. I'll be done in about 2 minutes. There are societal consequences, as we heard earlier today, to DNA. It's not done in a vacuum just with a crime lab. And what's truly happening is that much informative DNA evidence goes unused. There are these interim qualitative methods based on thresholds, consensus, whatever, where thresholds are applied because there were no computers or calculators that could do this. And in order to do anything at all, an analyst needed to be able to do something with the data. But the end result is that within cases and on investigational databases, these inclusion-based alleles, which have a very high false negative rate, so they don't detect what you want them to detect, and our studies with many groups shows that they lose about 6 orders of magnitude in a typical case, use alleles. Some national databases only use 2 alleles. They lose more information. CODIS uses more than 2 alleles. So it preserves a little more. It gets the full power of RMNE. But as we've read in recommendation number 1 from the ISFG paper by Gill et al., likelihood ratios are preferred to RMNE, and perhaps DNA databases should be built on the most informative way of preserving information instead of what's believed to be the least informative way of preserving information on the database. So the results of inaccurate, uninformative DNA interpretation, and if you want to read a controversial newsletter, I'll give you it's there. Go into the story. Not that controversial, but if you're invested in this stuff, it might be. DNA analysts do lose genotypes to the methods that they are required to apply. Many analysts don't want to be using these methods. Prosecutors lose criminal cases. Very importantly, for preventing crime, databases lose investigational leads, and the result is that people lose safety and they can lose their lives because we're not detecting and preventing the crime that the vision of DNA databases that came about 20 years ago was supposed to be doing for us. There is some consensus. Some of you may have been at the Australia meeting where Peter Gill, John Buckleton, Bruce Bedolia, and Angela Van Dahl all got together and had some disagreement about what is low template DNA, what are the interpretation guidelines, and I was asked to ask some questions afterwards, and I asked each of the 4 panelists 4 questions. I got 16 yeses and I stopped. A minor miracle. And here are the questions. Do you agree that DNA data is continuous and has random variation? Okay, you already agreed. Thresholds do not work for low template DNA. I think different people agreed for different reasons. Mathematical models can account for random variation. Unanimous consensus there. And the question of can objective computer interpretation inferring genotypes with probability be a good way forward for the next century? The answer was yes. So, in conclusion, our goal is strengthening forensic science. The systems that I work with satisfy the goals of being objective, reliable, and providing a consistent interpretation that can be reproduced. We spend most of our time now just doing validation studies and writing them up. 2 are published so far. And the goal is to preserve all available information from the scene of crime through the court, whether it's investigational databases or court reports, whatever. The approach is to use probabilistic genotypes, recognizing that the genotype is the summary statistic, the only summary statistic from DNA evidence that enters into the likelihood ratio. And the more informed those probabilities can be, the more we can preserve. We use, and other groups as well, use modern statistics and computation. And by using this type of system, forensics is relying on a solid mathematical foundation instead of on intuition or potential bias. Peter Gill talked about a gold standard. I'd suggest that systems that can extract, well, as far as we know, all the information out of the data might be a platinum standard for preserving DNA information and assessing the efficacy of other systems. And they move us beyond the less informative interim methods. If you have questions, my email's here. It's also, you'll get it from the conference as well. And if you'd like to learn more, there's part of our website that's completely dedicated. It's non-commercial. It's free. It's the papers, the courses that we've developed for people who want to learn about this, presentations, publications. In fact, there's so much on there that last week we put up a Getting Started link under information, so you'd have some roadmap of how to spend your first week instead of a day at a time, instead of wandering through the entire place. So thank you very much.
Thank you.